Good morning. Today I have prepared something good for you, and today I want to deal with HIV/AIDS cycle, HIV/AIDS life cycle. I don't want to go deep on the areas of HIV, but I want to specifically talk about the life cycle of HIV/AIDS. We still know that HIV/AIDS is a deadly disease, and we are supposed to stay safe. You're supposed to know how to be away from it. Uh, this is the way how HIV/AIDS, or the virus, enters your body. Once HIV enters the body, typically through sexual contact, or maybe blood exposure, or mother to child transmission, it seeks out a host cell to produce in order to produce. So it usually just enters your body, but it is finding a host cells, and these cells we call them CD4 cells and or CD4T cells, and sometimes this is white blood cell. And this is because the CD4 cells are the defense mechanism that defends your body. So once if it enters to the CD4 cells, it weakens the power of how to function. That's why you'll see someone becomes weak, because the defending system of his body or her body has been weakened. Therefore, I want to explain something under the life cycle of HIV/AIDS, and there are about 7 cycles, and I want you to be very keen when I am explaining about this thing. The first cycle is a viral attachment. A viral attachment. I've said that once if HIV enters the body, typically through sexual intercourse or sexual contact, or maybe blood pressure, blood exposure, or a blood transfusion, mother to child transfusion, it seeks out a host cell in order to reproduce. Okay, this virus needs somewhere where this fertile, so that it can keep on reproducing. The host cell, or the host in the case, is the CD4 T cells, and it's used to signal an immune defense. It's immune defense in your body. So this virus attacks these CD4 T cells so that it can weaken that immune system. So in order to infect this, HIV must attach itself by way of lock and key type system. The keys are proteins on the surface of HIV/AIDS, which attack to complementary protein on the CD4 cells, much in the way a key fits into a lock. This is what is known as a viral attachment. And a viral attachment can be blocked by an entry inhibitor class drug called transcriptase or Varroc. Okay, so there's a very hard terms on this drug, because we are talking about the HIV/AIDS and the drugs that can hinder that virus from entering your body. So this inhibitor, that is class drug called Silvestri or Varroc, it is very important, because once if you get that drug, it will block this virus from attachment. It will never attach in your CD4 cells. So the first cycle of the HIV/AIDS is a viral attachment. And then the second one is a binding and fusion. So once if that inhibitor, or maybe the Varroc inhibitor, is not applied, it means this virus will attach itself to the CD4 cells, and once if it is attached to the CD4 cells, it will bind and fuse into. Okay, so once attached to the HIV cell, or once it attached to the cell, HIV injects proteins of its own into the cellular or the cellular fluids or cytoplasm of the T cells. So that causes a fusion of the cell membrane to the outer envelope of the HIV version. So this is the stage known as viral fusion, and once it fused, the virus is able to enter the cell. Okay, I've explained the first cycle, that is viral attachment. So once if it attaches itself to the cells, now this virus fuse into the cell nucleus, and I've told you that this virus finds somewhere to reproduce. So therefore, if you allow it, if the person will not get this inhibitor, the viral inhibitor, it will end this person be affected, or this cycle will continue. So binding and fusion will take over, and I've said that once if it attaches to the cell, so HIV injects proteins of its own into the cellular fluids or the cytoplasm of that T cells. This causes a fusion of the cell membrane to the outer envelope. So you're supposed to know these things. Now there's kind of inhibitors that can prevent from this virus from binding and fusion, and an injectable HIV drugs called Usion is able to interfere with the viral fusion. So Usion is a drug that prevents the fusion of this virus to the cells. So you're supposed to be very careful when dealing with this. There are about 7 cycles that I want to explain, and I hope that everybody will understand about these things. And you know, everything needs some knowledge. If you apply knowledge here, you will realize that everything goes well. Now I explained about attaching and viral attachment and binding and fusion, and as we move on, we realize that there's some kind of other issues that you're supposed to understand them also. So from viral attachment and binding and fusion, that is viral encoding, HIV uses its genetic materials, that is RNA, to reproduce by hijacking the genetic machine of the host cells. In doing so, it can churn out multiple copies of itself. So it finds some kind of that genetic material, RNA, and if it gets an opportunity, it affects, or it enters to that, and it churns out and multiply copies of itself, and this process is called viral uncoating. Now the conversion of RNA to DNA, that is the building blocks for a new virus, cannot take place. Okay, you are supposed to apply some kind of inhibitors to the binding and fusion stage, so that you can prevent from these things to happen. So from the viral encoding, I've said viral encoding involves the genetic materials. The genetic materials whereby the RNA, that is the single in the material, it copies itself, and you realize you are getting the DNA of that kind of virus, that is some multiplication of itself. So you are supposed, you as a person, you're supposed to prevent this. You can start from the stage of binding and fusion to prevent this stage of viral encoding, so that you can prevent this virus from multiplying and generating some kind of copies. From viral encoding, we have transcription and translation. Viral transcription and translation. This once in the cell, the single strand array, as I explained under the viral encoding, this is single stranded array, that's genetic material of HIV, must be converted to the double-stranded DNA of the kind of virus. It accomplishes this with the help of the enzyme called reverse transcriptase. Sometimes this stage is already called reverse transcriptase, but you can say transcription or translation. I've said this single-stranded RNA, this genetic material, is supposed to be converted to a double-stranded, that is DNA of itself, to accomplish all this, and this it needs the help of an enzyme called reverse transcriptase. So reverse transcriptase uses building blocks from the T cells to literally transcribe the genetic materials in reverse from RNA to DNA. Once converted, the DNA, the genetic machine has the coding needed to enable viral replication. So drugs is supposed to be applied there. As we have seen under the first cycle, the first cycle is the viral attachment. We have seen that under the viral attachment, this virus finds somewhere to reproduce, and once if it gets a chance or opportunity, it enters and it finds somewhere to attach itself, and this is the cell, the CD4 T cells, which is the immune system of the body. And once it enters there through binding and fusion, and if there is no fusion drug applied, viral encoding may start to occur. And I've said that viral encoding is where the genetic material array reproduced by hijacking the genetic machine of the host cell, just hijacking because it can't rely on itself. It has to enter the cells and hijack it and start reproducing itself. So in doing so, it can churn out multiple copies of itself, and this is called viral encoding. So under transcription and translation, where we have said there's an enzyme called reverse transcriptase. So drugs supposed to be applied, and these drugs called reverse transcriptase inhibitor can block the process entirely. And this type of drug, nucleotide reverse transcriptase inhibitor, that is, you'll see NRTIS, which means nucleotide reverse transcriptase inhibitor. A nucleotide transcriptase inhibitor, sometimes NIRTI, that is nucleotide transcriptase inhibitor, and non-nucleotide reverse transcriptase inhibitor, that is NRTI, contains 4 inhibitors of the DNA. CHAM cannot be fully formed, and the replication is blocked. So under the transcription and translation, I've said that there is chamming and replication of all multiplied copies, the production of the copies of this kind of virus. So I've said that each stage has a drug to inhibit it. So I've said the drug that is applied under this stage of transcription and translation is a reverse transcriptase inhibitor, which can block the process entirely. And we have 3 types of this drug nucleotide reverse transcriptase inhibitor. That's the first one. The second one is nucleotide transcriptase inhibitor, and then nucleotide reverse transcriptase inhibitor, which contain fault mutation of the proteins which insert themselves into the developing DNA. In doing so, the double-stranded DNA CHAM cannot be fully formed. and replication was also blocked. So those are the things that you're supposed to understand. And as we move on, you'll see different stages. We have talked about viral attachment. We have talked about the viral encoding. We are now talking about transcription and translation. We have binding and fusion. They are supposed to be in order. Viral attachment, binding and fusion, viral encoding, transcription and translation. There is assembly and there is padding. So we are remaining with 2 to go, and this is assembly and the reverse. It's very simple. But the integration and assembly, those are the cycles that we are supposed to understand them. And this is integration, assembly, and the last one is padding. So in summary, we have talked about viral attachment, we have talked about the binding fusion. If you don't apply the fusion inhibitor, you will realize that this virus attaches itself into the cell CD4 T cells, and if there's nothing else applied, encoding is also going to take place, where this genetic material array it's going to be multiplied to DNA. Then you will come to the transcription and translation, whereby there is jamming and a blockage of this kind of CD cells will just keep on multiplying. There's several copies of this virus will be produced if you allow them to continue reproducing. So then there's integration. In order for HIV to hijack the host cell's genetic machine, it must integrate the newly formed DNA into the nucleus of the cell. Drugs called integrase inhibitor are highly capable of blocking the integration stage by blocking the integrase enzyme used to transfer the genetic materials. So we are supposed to understand this under integration. These drugs are called integrase inhibitors, supposed to be applied so that it can block this genetic material from continuing multiplying. Then we have the assembly, which is also a stage. I think the 6th stage and life cycle. So once integration has occurred, and HIV must manufacture proteins, building blocks that it uses to assemble new viruses. It does so with the proteins enzyme, which chooses or chops proteins into a smaller and then assemble the pieces into new fully formed HIV variations. A class of drugs called protein inhibitors or protease inhibitor can effect block the assembly process. So each stage I've said that this kind of drug that can be applied to block that stage to continue. And then the last one is budding stage, or maturation and budding. So if there is nothing will be applied, no drug will be applied from viral attachment to the maturation, realize now this virus become mature and you can't resist or you can't block it again. So when the variation are assembled, they go through the final stage in which the mature variation literally bounds from the infected host cell. Once released into the free circulation, this variation go on to infect other host cells and begin the replication cycle yet again. So this replication or the life cycle will continue. If there is no prevention measures applied, you will realize all the time each and every stage will go well because there's no any inhibitor applied. But under the budding, we have, these are not drugs that can prevent the maturation. You realize under this stage there is no drug that can prevent or can block this virus from multiplication, from multiplying itself. So you are supposed to avoid from viral attachment. Before viral infusion, you are supposed to get some of those drugs, evasion drugs, before the uncoating and integration assembly, before reaching the last stage, which the stage that cannot be prevented. So those are the life cycle of HIV/AIDS. Let's meet next lesson, which are going to explain more about HIV/AIDS and the drugs. I have to mention each drug and how this drug is applied in each stage so that we can understand. Thank you very much for sharing with me this stage. Thank you, and have a nice day.